Hello, so we're getting started. Thank you everyone for joining, and some of you have traveled very far. We really appreciate it. I particularly would like to thank a great working group and Holger and Jan for hosting this meeting. This is a very productive collaboration for us, and we are really excited to start this workshop. Let me just briefly frame what we're going to be working on this day today. So I will briefly introduce the pharmacology and toxicology concepts, existing mechanistic frameworks that have been established, and then talk about a little bit about systematic reviews and how systematic frameworks can help establish credibility and transparency of the new non-animal methods and mechanistic information. So the rest of the workshop is going to be moderated by Rob DeVries. Rob is pretty much put together the agenda working with the committee. So I'll hand it off after the introduction. So we have many challenges in integrating the new science in general in risk assessment in the regulatory environment. So one of the issues, one of the such challenges is cognitive bias. So and that quote about if you are a hammer, you treat everything as a nail. Apparently, the oldest quote I found was from science philosopher Abraham Maslow from 1966. The lack of confidence, that's another issue. How does the regulator know if the new information is reliable for their purposes? And of course, resistance to change. So the regulators really need a rigorous and reproducible framework for doing their assessments. So pharmacology, so who considers themselves toxicologists in the group? Okay, so we have about half. So the pharmacology is a branch of biology that studies drug action, right? And there's a definition of a drug. And that's the toxicology is a branch of biology that studies the effect of poisons or adverse effects on a biological system, on a body, right? And these are the pictures taken in Niagara by the lake this weekend. So I noticed that there is if there is a pesticide sprayed on some lawns, there is a notice with what class of pesticides it is. I haven't looked it up yet what it actually means, but it's interesting. So, and then there is a famous quote from Paracelsus, so poisons is in everything and no thing is without the poison. The dosage makes it either a poison or a remedy. So basically the difference between pharmacology and toxicology is in the dose, because almost anything can represent a hazard of some sort. It's just the dose that makes it different. So the mode of action or MOA frameworks in pharmacology have been around for a while. So every drug discovery program starts with identifying and validating a molecular target, whether it's a receptor-ligand interaction, DNA binding, or such things. Then in a higher level of organization, there are cellular effects that are happening, then the organ effects and the organism, right? And they each have specified steps. Many of them we have a very good understanding of, many of them we still don't. And finally, on the organism effect, in case of the pharmacology, we have a desirable effect on the body, and in case of the poison or the dose that's too high, we have adverse events. So the adverse outcome pathway framework that will be mentioned many times during this workshop, it's It is very similar to mode of action framework. It has been, for those of you who don't know, on OECD website there is a AOP wiki, and there are some quote-unquote validated AOPs that are already used by the community, including the regulators. So this framework consists of the molecular initiating event and a series of key events that eventually lead to adverse outcome, specific adverse outcome, right? It's a useful framework, but there are some problems with it. One of them is that biology is not linear, as we all know. Another one is gaps in our understanding of biology. There are many things that we still don't know. So can the systematic methods, including systematic maps and systematic review, help us solve these problems? So what is a systematic review? So it's many of you are very familiar with this. So it's a transparent, traceable, and reproducible summary of the results of primary studies, and that's a modification of the Cochrane definition. So the systematic reviews and systematic maps really provide that transparency and objectivity and comprehensiveness that's needed in the regulatory setting. The main users of this tool are regulators and policymakers for obvious reasons. And why? Because the regulators obviously need the reproducibility, traceability, and rigor in risk assessments. So the differences between a narrative review and a systematic review is mostly in the framework. So there are specific steps that systematic reviews go through. And again, most of you are familiar with this. The research question has to be specifically defined. It's usually framed as a PICO population, is defined exposure, comparator, and outcomes is specifically defined. The literature search is very explicit, with databases listed and named, and search being defined. The study selection, quality assessment of the studies is being done, and Andy Rooney is going to talk about this. And finally, the evidence synthesis, whether quantitative or qualitative, is performed at the end. So these are, again, the steps of the systematic review. So you identify a problem, or formulate a question, you pre-publish the protocol, the evidence is searched broadly as possible, and you use a validated risk of bias and study quality tool to assess the risk of systematic error or bias in the process. So toxicology is providing a unique challenge in this process. The systematic review and systematic maps approach came to us from clinical medicine, where you have different study populations, of course, different types of studies. However, it's not as diverse in terms of species and study designs as toxicology. So we are really, the systematic reviews here are relatively new. It's about a decade old. The randomized controlled human studies in toxicology are not typically possible because of ethical reasons. There are various designs of epidemiological observational studies, and there are animal and now in vitro, organ on the chip, computational, you name it. So we really need the tools that would allow us to systematically evaluate and integrate these new tools into regulatory assessments. So here is the possible vision for such framework. So the systematic approach can be used to define the AOP and then subsequently find the appropriate tests that correspond to specific molecular initiating events or key events in the process. So that's one approach. So you can identify the AOP pretending that you don't know biology. So right now we sort of do the AOPs are developed based on the current knowledge. This is what I know, and this is what I can report and submit to AOP Wiki, which is fine and which is a great start. But what if we didn't know? How would we go about this? And there are some tools, and Michelle Angrish from EPA is going to talk about this, about systematic maps. Then these AOPs could be developed into AOP-based search strings, which again could be used to systematically, specifically search for particular mechanistic tests. So what we need here is we need the tools to do this work, and again Michelle is going to talk about this. And for defining the chemical and biological, and especially chemical applicability domain, which we need the process, and we need the process also for regulatory case studies on how to validate such AOPs and tests. So the future of mechanism-based risk assessment is really, I think, is starting to, I think most people agree that we are going to rely more and more on automated tools, on text mining and machine learning, and semi-automated, eventually more and more automated search and literature screening, and eventually having this living systematic review that would be able to, we would be able to update at whenever we wish. Right? So this is a busy slide, but this is a timeline of workshops and international conferences and important publications that have been in this field leading up to this conference. And I'd like to mention the first international forum towards evidence-based toxicology that Dr. Thomas Hartung has convened in 2007 in Como, Italy. And then the important event is the EFSA is the first regulator, as far as I know, to adopt a systematic review and start integrating it into the process in 2010. Then NIEHS OHAT approach for integrating evidence and the risk of bias. The UCSF navigation guide for epidemiological studies. And then US EPA TASCA, which specifically names systematic review in the process. More recently, we just in 2017, we convened the colloquium on evidence integration in Lisbon jointly with EBTC and EFSA, where there are some problems that were identified and some solutions were proposed for working on priorities in the area of evidence integration, again focusing on this very diverse lines of evidence that we have in toxicology. Then there are two workshops that one just last week convened by National Academies of Sciences at the request of US EPA IRIS. The first one was on conducting systematic reviews of mechanistic data, and the second one on integrating the evidence. And again, we're still having a lot of problems in both areas, and particularly in evidence integration. So we need the tools to do that. And finally, there was the week before, we had Paul Whaley convene the editors, toxicology journal editors workshop in Durham on the quality of systematic reviews. As the tool is becoming more and more popular, there's more and more systematic reviews, and there was an agreement between the journal editors that there should be some tighter criteria for defining systematic reviews. So watch out for some publications coming out of that workshop. So I will start this meeting then and hand it off to Rob. So this is our morning program. So we'll have Holger giving a bit of an introduction to to grade for those of you who are not familiar. Again, Holger is our host here. Then we'll have Jan Brozek, also from McMaster University, who is going to talk about the modeled data and the certainty that we can have in the modeled data. Then Michelle Angrish from EPA on systematic maps and literature-driven AOPs, which I'm really excited to hear about. And Andy Rooney from OHAT, NTP on risk of bias appraisal of in vitro studies. And Patience, as a regulator from OECD, is going to talk about how they're tackling the AOP and what they want to get out of this systematic approach. And then we're going to break into the groups and work on specific problems and questions, and you were assigned those groups, so Rob will explain later on. So with this, I'd like to open this for a quick quick Q&A. If any questions arose, any thoughts, happy to start the discussion while we're waiting for Holger, because he's simultaneously starting another session in another room.
I have one quick comment. There's, a lot of material that you're presenting, and Hi, Andy Rooney, National Toxicology Program. And I just wanted to make one comment. In Katya's introduction, there's a lot of material there looking at the history and how we got here today, including a number of different workshops and many pieces on your history slide, or your timeline slide, were not even covered. And I think it's fantastic, and knowing many of those meetings and the progress that was made at them, that I'd like to see the list on the products that came out of these, because there's equally as many products and steps forward that have built off of these meetings. And I think that would be an important, concrete list for somebody else to develop. I'm not asking or offering to develop it, but just to emphasize that it hasn't been just meetings. It's been the progress that has been made.
Right, yeah, absolutely. And that's actually, they're color-coded. In green are the actual products and publications and tools and such things, and in yellow are the workshops on this slide. So, yeah. So we are waiting actually to hear. I think there's going to be a lot of products from this recent activities that we had as a community, yeah. Including long-awaited paper from Paul Whaley on biological plausibility.
Jan is Associate Professor at the Department of Health Research Methods, Evidence and Impact here at McMaster University, and is very active within GRADE. And he will be discussing how you could grade modeled data.
Okay, sorry, I will have to, I have a tendency to run around because I get excited when I do this, and I don't have the clicker, so I will have to come back to the computer, sorry. So very recently we met in Hamilton, well, very recently, maybe not, in 2017, and convened a workshop, and to address this issue, the certainty of model data. In other words, how much can we trust? How much can we believe in these, in the results of models in general? And we convened several, people from all modeling pathways and clinical and public health. I told you. Paths, and to have design a common, if not uniform, some kind of approach to how to actually assess this. How can we assess and communicate? How much can we trust? We believe the results of modeled evidence, and for the decision, really, of decision making, for the not decision, for the purpose of decision making in health-related disciplines in general. My potential conflicts of interest are that I work with the GREAT working group. The GREAT working group is an international group of, Katya just mentioned, of pretty much every methodology professionals, represented in that group that have anything to do with decision making in healthcare. So how to tell others and how to assess how sure we are about what we are telling others, etc. And I may say a lot of good things about that group, so that's my conflict. The Cochrane collaboration, I've been working with that group for another, 10 or more years, is a international organization, again, developing methods and standards for how to do systematic reviews, and we will be promoting systematic reviews here, so it might be a conflict, intellectual conflict as well. I've participated in many health care guidelines, developing them and developing methods for those, and advising some groups, international societies and governments about how to make recommendations or decisions. And I very recently, which means last week, I started, it is a financial conflict possibly, I started working with a company called Evidence Prime on something that Katya just mentioned, automation with machine learning of systematic reviews. So hopefully we will not have to do much of this. We got a nice grant, and I promise, if it's possible, in 3 years you will not do many things that we are doing today. I'd like to acknowledge, and it is the last slide of boring ones, the workshop participants because whatever I will say is their work and the great project group of modeling and many other people who were involved. It's not just me. For everybody in the group, I think it's pretty obvious. We are talking about models, but we decided to focus on these kinds of models. Formal rather than the back of the envelope. Quantitative, mathematical representations of the reality, and how to actually assess the certainty or the quality of these of the information coming out of these models. This is just a reminder. I think something that was very clear for us when we had the workshop is that the main difference is measured versus predicted results. And that's how we, at that time, started thinking about this and dividing these two types of evidence. But it's not, again, possibly helps thinking, but not necessarily very part of the approach that we are discussing. This is the first larger agreement that we reached within the workshop participants and group. Is really that the models may be very simple, and people agreed that this also is a model when we have some relative measure of some effect, and we multiply it by some number, which is the baseline risk in people. We got some other number which is not directly measured in any way, but is actually an approximation based on other two measurements. And anything in between up to the super sophisticated models of toxicology that and economic models and system dynamics in infectious diseases, et cetera, et cetera. So that was the first kind of agreement and we'll try to build on this unless we change our minds, thanks to you or other people who would contribute. It's a concept right now. It's not very detailed, but that's how we try to think about this. We also found out very quickly that we all come from all walks of life, and we talk to each other, and we cannot understand each other. I mean, we spend sometimes hours with colleagues, otherwise reasonably, I thought. Colleagues they couldn't get it. They couldn't understand me, and it was so frustrating. And they were also frustrated because I couldn't get them. And it turns out that we all, these things developed in parallel, but we use same words, but they mean different things. And we use different words for the same thing, etc., etc. So one of the things that we decided to do is actually to somehow agree on terminology. And maybe we will not agree ever, because standardization is the most difficult thing to do for humans. But at least we will have some translations and some, we use this word, this is what it means, and these are the possible words for this, right? So that we can, and we will still work on this. There are some examples of this work being already done and by WHO and other groups, but it needs a little bit more work clearly after this. The reason why we're talking about certainty of evidence is that we used to talk about quality of things, how good something is. Quality is a confidence in that thing, trustworthiness, whatever other word we will use. It has already been taken and actually meant something else for various people. So the only one that kind of stood the test is now the certainty. With all the limitation of this, and we are talking about certainty of evidence rather than quality of evidence or trustworthiness of evidence, etc., etc. Certainty, when we say that, we mean how much can we trust the results, how much the results that we have, information that we have, evidence that we have is actually helping us make a decision. There are 3 levels on which this may be assessed. So it's certainty of the inputs, certainty of the model itself, and certainty of the output. And when we talk about certainty of modeled evidence, we mean the last one. So that's also something that became very clear to us during these discussions, that people could not communicate somehow, and there was this inability to understand each other, because in certain fields, quality, certainty of modeled evidence actually meant certainty of inputs being modeled, so some information being modeled rather than being an object of modeling rather than the result of modeling. To distinguish this and being very clear what we are talking about, we try to talk about certainty of outputs, and that's what we talk about, and that's what's important for decision making. And in order to assess this, we need to know how certain we are about inputs and how certain we can be about the model. So certainty of modeled evidence is outputs. When we say that, that's what we mean in this particular context, acknowledging that there are different uses of this terminology. Katya mentioned the grade approach, and traditionally the grade approach. How many of you are actually familiar with the grade? Yeah, most of you. Okay, so I will not be spending too much time on this, but you know that there are certain things that may decrease or increase how much we believe something or trust the information, and there are the usual that we identified for other types of evidence or about interventions, diagnostic tests, prognostic information, etc., etc., economic information, that may reduce our certainty, and some that may increase our certainty. Each of these fields, if it's diagnosis or treatment or prognosis or risk assessment, which it will be assessed differently because different types of evidence are being assessed. And different problems may be affecting that evidence. But in general, conceptually, these are the things. There may be a risk of bias in this evidence. It may be what we call indirect or not applicable directly to a certain situation. Again, it doesn't matter if it's some kind of a treatment, a pill that people swallow, or the toxic gas that they inhale, or whatever else. Or the test that we do them, there may be not exactly the one that we are interested in. Inconsistency among different studies or different measurements of the same situation. Maybe just very few observations. It's just very not precise. We're not sure about the effect, how big the effect is. And then something that is called publication or reporting bias, which is pretty much we know or we suspect that some information has been collected and has been generated, but it's not available. So that's a bias because there may be a reason for which it is not available. Maybe it just didn't show things that people wanted to see. And there are some things that increase our certainty with our huge effects. Typical parachute example is an example here, right? We don't need experiments and other models or whatever to believe that parachutes actually work when you jump from an airplane. When we see the dose response, which means the more of the exposure, the more of the effect. And something that I will not go into, but it's the effect of residual confounding that would work against what we see is possibly also increasing our confidence. Maybe there's some more. We couldn't identify more yet, but that doesn't mean that there aren't. So if you have any ideas, ever, please let us know. So this is more or less. And we agree that all these are applicable to modeled evidence or evidence about outputs from the models. But of course, the way that it is assessed, the problems that will be there, et cetera, will be quite different from all the other types of evidence or bodies of evidence that we have, that we work with, and that remains to be done. How to do it, how to assess this, what to assess and how to assess it. General concepts are there. And this is the final proposal how to do it. So we would start. You don't see much, right? Okay, I'll make it larger. Is it visible? Is it okay enough now? I'm sorry, I'm just asking. Is it? Okay, thank you. So by formal modeling, we mean some kind of a mathematical quantitative modeling rather than back-on-the-envelope approach. So whether this is important, this is, if it's not needed, then people may forgo formal modeling. I understand that in many situations, or most situations, it actually is needed. The suggestion is to start with an existing systematic review or systematic search or systematic survey, people suggest to call it now, as Katya mentioned, of the existing models, of the existing meeting some very specific criteria that will help us answer the question that we are dealing with, the question at hand. So we would have to think of these criteria, the ideal model first, and then look for the models that are as close to this as possible. If the question is, can we find them? Well, if we don't find them, then the answer is obvious. We either develop our own model, or if we cannot do it, we unfortunately have to. We don't may, we just have to forego modeling, because if there is no model, we cannot do our own, that's it. If we can, then we would develop a new model and assess how, the certainty in the results of that model the same way as we would assess them for any other existing model. In beginning of this, there are certain assumptions in this whole pathway. And one of the assumptions here is that we actually are able to assess whether the new model will be any better than any existing model if we find them, right? So it's if we find some models, if there is no model, that's fine, but it's existing one. But if there are existing models, we would assess certainty of outputs, which is the model itself and the inputs that we mentioned, the criteria, the 5 that decrease the credibility and 3 that increase it. For each of these models, and we'll try to find a better one. One of them, we'll try to find one of them that is optimal, the best for our situation. The others are clearly not as close to what we would like, what we are modeling, the situation we are modeling, and this one. And we would try to find out, and it's more certain, better model. If yes, we can find it, then we would use it off the shelf or some adaptation. I mentioned that there is some assumption there. Doing our own models, if we cannot find any, if there are, some people suggested to start with your own model because it is always the best. So, that assumes that we actually can develop a model that will be better than existing ones, and two, that there is enough knowledge about the problem that we can actually tell that the new model would be any better than any other. That's why we are trying to start from systematic searching for existing models rather than suggesting that people develop their own model from the beginning. Theoretically, own model for this particular situations we're dealing with would be the best for this particular reason. If, in ideal world, if we knew everything about what's happening there, et cetera, but with the limited knowledge that we have, this might be actually a more efficient approach to this, because in many, and I guess toxicology is one of those very prominent, good examples when it's really not well known what's going on. That's what we will be talking about today, right? Lots of unknowns, the pathways that we really don't know much about, or are huge limitations of what we know. It will be hard to figure out which models, it is actually hard to figure out which of the models that we have is better. And so if it's possible to choose one optimal, we would use one. If it's not, if it's possible to adapt one that is not optimal, we would do this. If it's not, there is no single one that's better, there is no possibility of adaptation of one model, then again, there is a question of being able to develop, and if it's possible, yes. And then if there is just one model, that's it. Sorry, I think I repeated something here. Okay, no, okay. So the 3 steps, choose the best model if it's possible. If not, adapt. If not, if it's just one model, use that model if possible. If it cannot tell which of the models is best, I guess that's the situation in toxicology many times, that we run many models because we just don't know which one of them is better. If I understand correctly, and correct me if that's not true. Then there are several ways of trying to figure out across models which of them is better, and then we would assess the certainty across all the included models and how much can we trust the information across all these models and the final results. So that's more or less the approach that we have right now. We'll be discussing more detail tomorrow. There is a paper that we are drafting, and many of you have already seen and provided feedback. Thank you. And the next steps is really, how do we do it? So this is kind of a conceptual framework because of efficiency, because of the real world situation, how we would do this, but then in general, but then how to assess actually each of these things, how to choose this one model that is the best, what makes it the best, and how actually assess the evidence across models is something to be worked on. So it's just a summary. Simple and sophisticated are all models, and there are 3 scenarios. New model, developing a new model, theoretically the best, but practically may not be possible. Use off-the-shelf or adapt existing model, or have to assess how good it is across models. And the 4th is foregoing modeling.
Thank you very much, Jan.
Sorry, I think.
We have room for one clarifying question, yeah? Just a quick question?
Oh, clarifying question?
Yes.
Okay, I'll try it. Clarifying.
Yeah, okay.
Can you go back to your slide? The first time that you talked about sort of the
Which one, sorry?
The first big purple box.
The first forward?
The single study, yeah, that one. So that box that's pointing next up, you can't read the text right now.
I know.
But it talks about, so here you're talking about individual models, right? Looking at, and so I was wondering, and then you see sort of the grade factors, and I was wondering, it sounds like you're thinking about applying the grade factors, which is typically more across the body of evidence, to individual models. And I was wondering about that, you know, like things like consistency, and if you're just probing one model, about whether you think grade really sort of applies at that phase versus down below. And it could be that when you do this, you identify only one model suitable, and then it does apply. But I was wondering if at that phase you think grade applies.
Well, you could think that it applies in a way that if you perform a search for many, like a systematic. I'm coming from a clinical work, so for me, not models but single studies are easier to think about. So, and we systematically search for studies, but there's just one of them, and we find one, and we still can apply the grade thinking, the approach to that one study. You know, the risk of bias in the study is the directness of the information, so applicability and all the concepts related to this just cannot be inconsistent with itself. So inconsistency is kind of irrelevant if you have one, and then everything else applies the same.
Your next step is to say either one or multiple, and it still seems like this is still upstream from selling on about one model.
So again, if you would do having your criteria of what is really fitting your situation, that's how I tend to think about it. And please chime in if it's not. If you would find a one study or a model, that's, you know, real life study or a model that is clearly more direct and better than the others, the question is, should we be even looking at the others that we found, right? Because they either don't model the situation as well as this one, or there are many problems with them, various reasons. And we sometimes do this with normal studies and systematic reviews. Yes, we find more, but we narrow ourselves and we say, okay, this is really the evidence we believe the most when making a decision, that one.
Okay, we have to leave it at this. There will be a general discussion for all plenary sessions, so please keep your questions for that. We can move to the next speaker, Holger.
Welcome to beautiful Hamilton. There's a parallel meeting ongoing on non-randomized studies as well, and I did my little introduction there, and that's why I was a little bit late, but really great that you could all be here. My goal for today is to provide a little bit of an overview of where we stand, give you a little bit of background about the workings of the GRADE working group, and then try to tie this back into what Jan has talked about. Before I do that, I think, Rob, you haven't talked about logistics at this point, right? That's perfectly fine. We don't have to do that, but we'll have time to provide you with some more information. All that I can suggest to you at this point is that use the lunch break to walk around on campus. The campus is actually pretty nice, and the weather is good because it's going to rain tomorrow. So use the opportunity today. It's going to be nice again on Friday. Okay, so and the other very important logistical item is, for those of you who come to I hope that everybody comes to the actual main GRADE meeting tomorrow, that meeting is in a different spot. So this is our main campus. We have a downtown campus. It's a nicer conference facility. It lends itself more to small group work, and that's why the other meeting is then tomorrow and the day after tomorrow there. It's a bit harder to book. And even if we started almost a year ago or something like that planning this, it was booked for today. That's why we have the two facilities. That's just by means of introduction. And obviously, thank you. There's a lot of people to thank you. First of all, Rob and Katya for setting this up and also supporting the meeting. Same is true for NTP that has sponsored some meeting attendees rather. for tomorrow that then led to being able to attend today. So the evidence-based toxicology group needs to be thanked, and Antoinette and Camilla and Anita who help with the logistics. That's really important. That's always important. So I'll start with providing you a little bit of an overview. These are my disclosures. One thing that is important is that grade hasn't appeared anywhere on this agenda. There's not that's very unusual. In the entire table of contents, the word grade doesn't appear, but you are here in the context of a grade working group meeting. I wanted to tell you this in case that you didn't realize that. The whole connection actually came about through some of the work that Jan has been presenting, that is really bringing some clarity to rating the certainty of evidence. Grade started about, for those of you who haven't attended any of these meetings, started about 20 years ago with the goal to bring some clarity to what I will explain in some more detail, clarity around assessing when we can have certainty in the available evidence. And that evidence, as we've learned over that type of evidence, or the type of evidence as we've learned over the past few years, is expanding. So we started 20 years ago. After about 20 years of confusion, lots of organizations are using it, have participated in developing it, and GRADE is focused on systematic reviews, and I think we will hear more about the National Academy of Sciences workshops on systematic reviews that Chris sponsored with EPA, and a follow-up workshop. So systematic reviews, that's where GRADE applies. It's the method for Cochrane, health technology assessment, and also development of recommendations. It's a big group, international, diverse contributors, different backgrounds. Right from the start, we had lots of public health folks involved. When I started, was still a trained classic epidemiologist. I think my move to clinical epidemiology happened after I got involved with GRADE, quite interestingly, but there's reasons for that. You know, we've been very successful with our publications. There's quite a few publications that Rebecca and other folks in the environmental health project group led that are now coming out in Paul's journals, as I call it. The Paul's journal, as I call it, but it's not Paul's journal, you know, it's yeah. Anyways, you can talk to Paul about what the right term for that journal is. But so quite successful, and we have a handbook and we have an IT publication that we will hear about. GRADE has just to put this in context for those of you who are not familiar with it, GRADE has now 16 centers and networks around the globe, really. So we are on all inhabited continents, as I call it, although Antarctica, apparently there's a few people surviving there as well. We will never make it there. Maybe our work will be applied there anyway. So there are 16 centers and networks around the globe that basically support those who want to apply the methodology. I mentioned over 100 organizations. There are project groups. This is from our website that anybody can get involved with, and the environmental health group is one of them. So I wanted to draw the connection, and some of you will have heard me say a few of the things that I'm going to emphasize, but I wanted to draw the connection back to the AOPs, which has become the primary theme for this meeting today, okay? I know nothing about AOPs. I did a lot of basic science in my postdoc, all the, you know, geographical directions, northern, western, southern, blots, and all this kind of stuff, PCR, animal experiments, but AOPs were not on the map back then, okay? So this is, I don't know if there are other papers, but I use this paper as an example. I think providing, you know, written from toxicology, providing some sort of overview, and that's, I think, what we are dealing with, right? That's what we are talking about. And as you can see in this introductory paper, Tool in Toxicology, there is mentioning actually that development of AOPs ideally complies with OECD guidelines. This also holds true for AOP evaluation, which includes consideration of the Bradford-Hill criteria. There they are, my favorite argument. So the Bradford-Hill criteria to then assess the evidence and what people call here weight of evidence, which is just another term for strength or quality or certainty of the evidence, the way that we look at it. So I wanted to emphasize that in this context, GRADE actually came from epidemiology and from these concepts around Bradford-Hill. So this is the original paper by Bradford-Hill, basically talking about certainty and causation. And this over time led to quite some related work in the clinical context, so that certainty of causation, the first rating system that was used in the context of making recommendations was actually developed by the Canadian Task Force on the Periodic Health Examination, and it was people right here at McMaster who were playing a critical role in this. Dave Sackett was one of the people then bringing this into the clinical arena, but it really came from kind of preventive medicine, public health. And you know, the idea there was already to put evidence together, synthesize evidence. So the rating system ended up with Roman levels of evidence, Roman number labeled levels of evidence that were focused on bringing different pieces of evidence together, and included an assessment of things like risk of bias, for instance, by saying, you know, the evidence from randomized controlled trials is better than the evidence from observational studies, that then led to recommendations. And GRADE is very much a development of this work that was published now over 40 years ago. So I wanted to emphasize this point because it's quite relevant. Bradford-Hill did his work in the 50s and 60s. Then, as we are doing it now, Dave Sackett and colleagues picked up on this work that was done by Bradford-Hill. And Bradford-Hill was an individual, obviously. Dave Sackett was a group of people, and we think that GRADE is a community that actually can address some of these issues that we are facing here. How certain, as we can be, as was described in this introductory paper on AOPs, how certain can we be in this evidence that we have, in this adverse outcome pathway that gets developed in different ways. The other thing that I think is really important, we confuse it all the time, is the distinction between association and causation. Bradford-Hill very much focused on causation. I just wanted to make this point. Again, this is unfortunately shifted. I cannot believe it, that happens after years and years and years of Microsoft and whatever Apple working together. Doesn't seem completely working out, and it seems to be always my slides. Anyways, so just to distinguish the two things, one is, you know, you might ask a question that could look at an association. We used the ethylene oxide example in one of the National Academy of Sciences workshops. So you could ask the question, in people or humans exposed to ethylene oxide, which is a very common byproduct of that is used in various industries and in the including in the health industry. What is the impact of exposure to ethylene oxide compared to no exposure, different levels of exposure, and so on the occurrence of cancer? And another question is an intervention question and a decision question, and that relates to whether or not regulation of the exposure to ethylene oxide, for instance, a reduction compared to no regulation, has an impact on the various outcomes that we are interested in, and that is a PICO as opposed to a PECO question. Okay, so just coming back to Bradford-Hill for a second, and I like to refer back to this paper because we started thinking about Bradford-Hill criteria about 10 years ago in the context of GRADE, because we were asked, where, you know, how have you evolved from Bradford-Hill into your certainty domains? And the first thing is, what is the certainty of the confidence in the estimates or the confidence in the association? And there was a big question in GRADE, and that was, what is our starting point in terms of the certainty? Is it, we're looking at a body of evidence and we are trying to evaluate or find out is there anything in that body of evidence that makes us confident that there truly is an association and a particular causation? Or do we start by saying, okay, we are rather looking for reasons for lowering our confidence because there is a body of evidence, and GREAT has chosen that latter approach where we typically say, okay, we are pretty confident, we are looking for kind of reasons because it's much easier to do. We found after lots of elaboration, it's much easier to do. We are looking for reasons for lowering our certainty in the majority of cases. And that was somewhat different from the Bradford-Hill criteria or Bradford-Hill considerations, as they are correctly called, where we're looking for reasons to increase our certainty. The problem was that obviously over time the Bradford-Hill criteria or considerations had become outdated. For instance, publication bias was known when Bradford-Hill put his considerations together, but he simply ignored it. So the characteristics or considerations we found were really not complete. That was one of the main reasons for thinking, okay, and also Dave Seckett's reasons, there needs to be elaboration, evolvement of that method. They were not properly operationalized, so issues around imprecision were not very well described. They were not completely thought through. So for instance, issues around associations and interventions, so there are lots of how to apply them to other questions around prognosis and tests and so on. So we felt that it was really not fit for what follows even in the context of exposure assessment, policy and interventions. So I will skip over that and then basically just say that at this point we've given consideration to all of these criteria that continue to appear in the literature, including for the AOP, but we think that it's time to stop thinking about them in the way that their considerations in the way that we thought about them in the past, because there are gaps. We've then said, okay, for those of you who have difficulties letting go, here is how to think about the Bradford-Hill criteria in the context of GREAT, and how do these things relate to each other. I will say, so this is from 2010, through my interaction with Chris, with Andy, and the rest of many of you here in the room, our thinking has evolved quite a bit around what needs to be done around some of the domains in GREAT, in particular indirectness. I think there's a ton of work that needs to be done, and to me the AOPs very much relate to that indirectness domain, and they relate to considerations around biological plausibility, which I will spare you now, but I'm sure I will come back to at some point when that comes up in one of the conversations. So again, as I said before, not everything made sense in that context. 50 years have passed at least. Bradford-Hill was one person. Spitzer, Sackett, and the colleagues who did their levels of evidence were a few people in the group. And we have made this a community to evolve this further. So Jan already took you through, and this is why this is actually quite good that I don't have to take it through again. This is what we are doing when we assess the certainty of the evidence. The criteria or the considerations from Bradford-Hill are included here, and we go beyond them. And as I said, and this is probably also addressing part of Chris's comment that I think is something that we should be discussing over the next 3 days, with all of GREAT provides probably in the context of modeling also the considerations around the domains, but not the operationalization. And I think your question really had to do in part with how are we going to operationalize the general domain of indirectness, even in a single model or in a single question, in a single study or across studies. It applies to both, I think to both types of questions, but we need to operationalize them. So GREAT thinks about assessing single questions, assessing the confidence in an estimate of the effect, single studies, but ends up typically with a rating of the body of evidence by outcome. And this may be a single study at the end. If only one study fulfills the criteria, or it may be a single model. And then GRADE also deals with the decision-making part through its use of the evidence decision frameworks, which we will not talk about here. I think we will only talk about the certainty of the evidence. The fortunate thing in terms of GRADE for many of the questions that we have been addressing in the past is that there has been more work on risk of bias assessment in this for most of the questions that we are addressing. That has made the work easier. I think in the adverse outcome pathway, the AOP world, this is much harder because it hasn't been figured out as well, but needs to be done. But I wanted to leave you with one thought in the context of what I said before, what I found in the context of AOPs. So again, this is being mixed up. But if you think of a sensible question related to a specific exposure, and I use this example again, so the question being in humans, what is the impact of ethylene oxide exposure compared with no exposure to ethylene oxide on breast cancer? So in other words, does it cause breast cancer? How large is the risk? If you think of that, my question is for you, if the AOPs do nothing else but help us ask the question. Okay, so keep that in mind. Do nothing else but help us ask whether or not this is an appropriate question. Okay, that's one way of thinking about it. Okay, then you can do your evidence, the certainty of the evidence assessment, but is it only, and many of you have seen this, and I'm still looking for somebody to put this into a nice graphical display, okay, if there's anybody with talent in the room. Is it at the end that we are looking at this population, the exposure, and the outcome, where the population is humans or animals, where we're looking at, mechanistic evidence, I will just put it as such, that helps us to extrapolate, for instance, from animals to humans? Is it that we are looking at some of the mechanistic evidence and possibly also the adverse outcome pathways to let us understand whether or not an exposure, the way that we can measure it or that we do it in the lab, is actually the type of exposure that we are interested in, I will not use the word real world because everything is the real world. It's not a great term, but in the for the actual population or question that we're interested in. We can use the same approach to say, can we actually model the comparator appropriately? And as you know, we've done this work with Paul and Chris and Rebecca trying to identify how to phrase questions appropriately in this context of exposure and outcomes. So does it help us to identify whether or not an outcome is actually a direct outcome or a surrogate? And at the end, again, sorry for the mix-up there, is it, are we just exploring whether or not there is a direct connection between the exposure and the outcome, which would then mean, again, on the basis of systematic review and after judgments of indirectness, which would then mean that perhaps it only helps us to pick the right question. And the next question then is, how certain do we need to be in the right question to be asked in order to move to human studies or evaluate human evidence? So that's my thoughts on adverse outcome pathways. Mechanistic evidence, so it's mixed with biological plausibility in the literature as well. This may not be something that you are mixing up, but certainly others have been mixing up based on my reading, and I think it would be good if we brought some clarity to this, if we start thinking about, assessing the certainty of the evidence around that. That's it.
Thanks a lot, Holger. So Michelle is a toxicologist at EPA, at the IRIS program, and she'll be introducing how to build AOPs in an evidence-based way.
Thank you. Okay, thank you for inviting me, and thank you for everyone for being here. So we're going to get a little bit more nitty-gritty into the details of adverse outcome pathways, and in kind of the direction that I think that we're thinking about moving for further developing AOPs is taking a lot of the evidence that we've been gathering and all the literature that all the information, literature, stuff we're extracting out of it, and kind of framing that in an adverse outcome pathway so that we can begin to identify areas where in vitro tests or alternative methods and may align and be informing some of the events. So briefly, these are my own views and have nothing to do with US EPA, sort of. Okay, so what is the challenge that we're talking about here, and why might adverse outcome pathways and systematic maps be helpful? And so the challenge is that interpreting mechanistic information in the context of literature-based chemical assessments is kind of difficult because it goes back to where do you align this? How is it important? Is it actually informative for the particular assessment? And why is this difficult? And it's difficult because the identifying pathways that are actually causal are kind of difficult to find, and they also need to be grounded in an evidence-based way. And so the idea here is that a possible solution to this problem is to leverage systematic mapping methods for a more data-driven approach, so expert-guided and data-driven approach to evidence integration. And then adverse outcome pathways, I don't really care what you want to call it, but adverse outcome pathways exist. And I'll show you some of the databases and tools surrounding that, but they provide a very useful framework for organizing that evidence. Okay, so real quick, this is kind of what you're in store for. I'm going to do a quick adverse outcome pathway primer, and then we'll discuss a little bit literature-based chemical assessments, evidence maps, and then I'll show you an example of how you would apply all of those to align information to an adverse outcome pathway. All right, so the adverse outcome pathway is basically a construct for framing the evidence, and this is essentially what it looks like. And it, I'm not going to read this to you, but you can read it, and hopefully I think the slides may be available. I have some useful links included in this presentation. But it was kind of born out of the ecotoxicology world to study population level effects. So what you have is an initial event where you have some sort of exposure, and then these events kind of progress through biology to arrive at an adverse outcome. And so this is the general framework where you have properties of chemicals. There's a toxic, well, it doesn't have to be a toxicant. It could be some stimulus that leads to a molecular interaction, a cellular response, organ level response, an organism response, and then potentially even a population response. And so we use words to talk about these events within an adverse outcome pathway framework. And so these are the events and words that we use, and there's definitions for these words. And so I look forward to the breakout session later today, and we'll discuss some of these words and the meaning behind them. And so I want to mention first that we have a molecular initiating event and then several key events in between leading to an adverse outcome. And all of these events are connected by key event relationships. And so it's a lot of the evidence goes into supporting that, if you can think of it as dominoes. So if you have an event being impacted, it kind of tip over, right, and then fall on all of the other events, eventually leading to an adverse outcome. Now, I cannot stress enough to not think about adverse outcome pathways as linear. They are not linear. They're often depicted linear because it's easy to draw a straight line in a 2D space. And normally when we're presenting things about adverse outcome pathways, it's on a piece of paper in a presentation. So as we heard earlier, biology is a network of things, and so adverse outcome pathways are also a network of things. And so this is on the bottom is the network representation, where you have multiple events and they can take different paths but arrive at the same outcome. It's kind of like driving, right? I mean, there's many different ways to get to the same location. Same with biology. There's often many different ways to arrive at the same outcome. Okay, so there's a little bit, there are several actually very useful databases and tools supporting AOP development, and so the AOP wiki is kind of like the sandbox for folks to begin developing their adverse outcome pathways. And so the really cool thing about adverse outcome pathways is that it is a wiki. I mean, anybody from the community can begin developing adverse outcome pathways. And so, it uses crowdsourcing essentially to collect knowledge. And it's in an interactive space on the wiki. And so there's a link on here. You can go to the AOP wiki and kind of explore and look around. It may not be super meaningful until you go to the OECD website and take a look at, the handbook or just a little bit more general information about adverse outcome pathways. But there's a lot of information already existing within the AOP wiki, and so you can go in and look for a particular event and see if it already exists. It uses a template form to kind of guide users as they begin developing. So there's a lot of information already existing on how to actually develop the AOP, what the organization should be, and even the words. Like the words are really important. We heard Jan mention earlier that we all use different words to say the exact same things, and people end up putting different words to say the exact same things in the wiki, and so it can get super messy. Therefore, it's really important to use harmonized or somewhat of a consistent, I don't want to say harmonized, but a consistent terminology to avoid duplication of events within the wiki. Some other useful databases and tools include the Adverse Outcome Pathway Knowledge Base and the Effectopedia. So and again, these are links that will take you to the information that I'm talking about. But the AOP portal is kind of a neat place to go in and see what events exist, and I think that this is the one where you get like the visual display, and you can go in and really interrogate the different events and components within an adverse outcome pathway. And Effectopedia gives you, you can search for a specific key event and get an idea of, how things are connected in the AOP space. And so these are going to be a little bit more, these are more for folks who are more familiar with adverse outcome pathways, but you can go in there and kind of play around and look at some of the information that's already existing. Okay, so I talked a little bit about the crowdsourcing and whatnot, and so you may be thinking, oh boy, if anybody can put an adverse outcome pathway in there, how do we know that it's any good? Well, it takes a little while to develop an adverse outcome pathway, and the way it is right now, it's not always easy to find all of the evidence, I think, that's supporting the events within an adverse outcome pathway. And a lot of the information is kind of missing. So we already have a lot of expert-derived adverse outcome pathways, and so you end up with something kind of like this, where this isn't from the wiki, but you have some events connected where these would be key events. So let's say this is an event where you have arsenic exposure, and it leads to changes in DNA methylation, DNA damage, and cancer. And so, I don't know why I'm looking up there, I can look here too. And so then the evidence supporting those different events are given in this table format. But this was not systematic by any means, and so there might be a ton of missing information. And what about other pathways that might be important to this cause and effect network that are missing just because the expert didn't know about them? And so that's where the whole systematic review methods can really help us in understanding environmental health science questions. And so what we've been doing since Chris came aboard more recently is incorporating systematic review methods into our processes. And so it's not that we're always doing a full-blown systematic review on everything, but we really try to use the methods so that all the decisions made from start to finish are really clear and transparent. So what we've been kind of borrowing from the systematic review process, because honestly we just don't have the resources to complete systematic reviews for all of the projects that we're working on, and it's not always that appropriate, but we can still borrow from the techniques and methods and apply those during some of our projects. So here's an example of how some systematic review methods and systematic mapping may help support or provide more evidence in an adverse outcome pathway that was previously derived by an expert. So the idea here is, okay, well, we've got a ton of literature. I'm interested in arsenic exposure, and I'm interested in the outcome cancer. So we can use those. We can use those two words to start bounding the literature. Like maybe we have 50,000 studies, and we can identify all the studies that are involved in cancer. So right here it's just showing a pie, and then only look at that 1945. And then if you try to bin a little bit further by DNA methylation, it begins to reduce the burden even further. And so here what I'm showing is kind of that process in a little cartoon where you have all of your studies. You begin bounding that literature by the exposure and the outcome cancer. So now I have fewer studies that I have to begin looking through, and maybe I'm only interested in human and animal evidence to begin, and so I'll take all the in vitro stuff and just tag it so I can look at it later, which is important, especially in the context of today's meeting, because we're thinking about, well, how would we get at that in vitro evidence? And it's not always clear from in vitro studies, that it might be important for the pathway that you're looking at. So we'll put that in a bag and kind of save it for later. So next we want to subset a little bit further, because we know that DNA methylation is really important in this particular pathway. So now I've subsetted the studies by arsenic exposure, cancer, and DNA methylation. And I'm going to begin to extract information from those particular studies that might be, helpful in identifying, areas where, where is the literature mapping. So what am I trying to say here? So we already have this expert-driven guided AOP. We can kind of use that as some of the, to guide some of the data that we're extracting in this process to identify, what might be some of the mechanistic underpinnings involved in this pathway. And so we have some really useful frameworks for helping us determine which types of information that we want to begin extracting. And so this is kind of a busy slide, but it's really getting at, when you're thinking about extracting the information, it's really important to think about, to be strategic in how you're going to represent that information, especially if you want to be able to use it, align it to an adverse outcome pathway, which actually has, characteristics. So there's terminology existing within the adverse outcome pathway here. So, we have a level of organization. There's different events and components in the context. So in the context here is what is the, are we talking about it is it a biomarker measurement, like did it come from blood, or is it a change in gene expression? This is really important when you start to align the evidence, because a change in gene expression doesn't always correlate to change in blood, and vice versa. So you need to maintain the context as you're moving through. And so this figure is kind of getting at that, the words and terms kind of thing, which keeps coming around. It's actually really important. And so I stole this. Oh, I didn't steal this one, but this is kind of what it would look like in the context of the epigenetic adverse outcome pathway that I previously showed. So cancer would be, it could be a key event, or it could be, I think this is supposed to say adverse outcome, sorry about that. And the key event here would be decreased P53, so it's a tumor P53 tumor suppressor protein. And so if it's decreased, then you have increased mutation, which could lead to cancer. And so then the other events might be right here. It's a component would be a process, so it's DNA demethylation. The object that's acting on is the DNA, and the context is molecular domain. So it's not blood. Here we're talking about molecular, so it's changes in DNA methylation levels. And so these are some types of information that's really important to capture, particularly if you're looking for in vitro assays that might be informative, or if you're looking, to maybe replace some of the events that were previously measured in animals by an in vitro test. Because the whole idea here is that if you have something happening at a particular level to cause this event, you might be able to infer that the outcome may occur. Okay, and so this is the slide that I stole from Steve. But I thought that it was really neat to kind of get at the whole idea that you have these molecular initiating events and you have these early key events. And so here's where we're really thinking in the context of developing assays that might be replacing or maybe not replacing, but be informative for some of those early events, and therefore maybe if you have a bunch of enough assays that provide evidence sufficient, then you would be able to infer that a downstream event's going to occur. And this is usually bolstered by epidemiology data at a higher level, so like an organism level. Whereas I think a thing that's kind of more up and coming is the molecular epidemiology data, so just biomarker measurements from people and such. And so I think that that's kind of mostly what I wanted to say, and I hope that it made sense. But there are many folks who helped me out with this, and so thank you.
And then I'd like to introduce our first speaker after the break, Andy Rooney, who is the acting director of the Office of Health Assessment, so OHAT, at the National Toxicology Program. Andy?
Thank you. Okay, in the theme, or the shifting themes as we're covering our topic base this morning, I'm going to focus in on study quality or risk of bias appraisal for in vitro studies, which obviously are part of, but not all of, the mechanistic studies that you may consider within an adverse outcome pathway. So I'd like to start by defining some of the terms or considerations in a critical appraisal process. And so there's the critical appraisal of internal validity, also called risk of bias, basically whether or not the study design and conduct may bias the results, an assessment of external validity, also called applicability, generalizability, a number of other terms, but basically this is covering the extent to which the study addresses the research question or the review question. And then reporting quality. You can see on my little building blocks on the right-hand slide here that I put reporting quality at the bottom. This is one of the foundations. This is the adequacy of reporting of an evaluation study, design, conduct, and results. In other words, if they don't report it, you can't determine whether or not they did it adequately or appropriately for your research question. And so it's definitely one aspect of the study quality or the critical appraisal. It may be addressed independently or it may be addressed as part of your overall process for risk of bias and internal validity. And then sensitivity is another concept. Whether or not the study design and conduct impacts the ability of that study to detect an effect. In other words, if they only tested high doses and you're interested in a low dose effect, it's not going to be relevant to your research question. If they only tested really low doses that you know are not going to be effective at producing an effect, then again, that study doesn't have the sensitivity for determining whether or not the chemical might be toxic. So within all of those, let's step back on those terms and then ask where are the in vitro studies, because I'm talking about two things today, about the mechanistic data as well as the in vitro studies. And mechanistic data can come from a wide variety of sources. It's generally focused on upstream indicators, away from the health effect or the disease phenotype. It may be part of a larger study that is associated or focused on the health effect, but it has other aspects in the study which include this mechanistic information, metabolism, cellular and biochemical or molecular events, along with the AOP pathway. And that could include a wide variety of study types. Your animal and human in vivo studies, in vitro studies, in animals, in human cells, and also in silico or the model data. This is the slide to address everything that came before, I guess. And then in terms of assessing risk of bias, you can use the existing approaches for assessing risk of bias for mechanistic studies with an in vivo study design. They can be assessed with the tools appropriately for the animal human models. But the in vitro studies and the focus of this talk moving forward, there's a question on how we should approach those or how we possibly could approach those studies in terms of risk of bias. And so there's various, again, starting from the top and extending the focus towards
in vitro studies. There's tools that have been established for a long time to evaluate human studies. Obviously, the randomized controlled trials, and then various approaches for addressing observational human studies. There's an ongoing question there, but the tools are well developed and they're moving forward and being refined. There's a number of tools to evaluate experimental animal studies. The CIRCLE tool, the navigation guide approach, the NTP, OHAT approach. The EPA has a methodology for assessing experimental animal studies. And then, again, building down, there's emerging approaches for addressing in vitro studies. I don't have ToxR tool on here, but that's an approach that's largely looking at reporting rather than the study quality, and it doesn't focus on internal validity. But the NTP approach that I'll present here, we've extended the approach that we applied to experimental animal studies to address in vitro study designs. There's a SIRAP, or the Science and Risk Assessment and Policy, that has a method that they've extended to the in vitro approach, and they're currently pilot testing. If you might want to try that out or check out their work in progress and have some impact on how that approach might be refined. I put the EPA IRIS handbook in here because they have an approach for how they handle mechanistic studies. They may not have a prescribed method in the current version of their handbook for how they address these in vitro studies, but they have an approach for evaluating the entire database. And then also to look at current ongoing efforts, and somebody asked me, where is my paper? There's an ongoing systematic review of critical appraisal tools that I'm part of at NTP and collaborating with EBTC to look at the ways that various published resources have evaluated in vitro studies, and to be able to synthesize those within a risk of bias context, as well as other aspects that might be beyond that. So starting with this OHAT approach, what I'd like to do is step you through our method, and that we have a parallel approach to addressing risk of bias across the evidence streams. So study design determines which questions are applicable, but we start with a set of 10 questions and an other question applied to all the different study designs with the criteria that differ as appropriate for the individual study designs. The evaluation is endpoint specific, so we've done multiple times within a study for different endpoints, and the rating is on a 4-point scale. It's established and published for human studies and animal toxicology studies, but we also did an approach for in vitro studies. And this is the context we did it within. There's 11 questions, again, the 10 basic questions, and the 11th one is, are there other threats to internal validity? And study design determines which of these apply. If you look at experimental designs, such as a randomized controlled trial or experimental animal studies, they would have the question for, was administered dose or exposure level adequately randomized, and it would be applicable to that design, but not to the observational studies. And similarly, question 4 on confounding is applied to all the observational studies and only applied to an experimental study on an optional basis. So that's what I mean by study design determines which of the questions apply. And then within that, the questions are not refined, but the criteria that define each of the ratings on terms of what aspects of study design, conduct, and reporting are required to reach that rating. What do you look for when you're evaluating a study? What distinguishes among the 4 risk of bias ratings? Those are the criteria that are defined extensively based on study design and the different evidence streams. And what we did is we took the experimental animal approach and extended it to explore whether or not that concept could be used for in vitro studies. And it is successful, but we open on that, how successful was it and what else can we do moving forward. So we haven't released it as this is the approach we would recommend for evaluating in vitro study quality. And let me go a little bit more detail on that. In adapting that criteria, we brought together experts from NTP that work in vitro and model data, both the NTP staff and contractors, and we introduce them to the concepts of risk of bias as opposed to how they might be evaluating study quality before that point. There's certain things that the in vitro methodologist might be looking for in study quality that wouldn't fit within a risk of bias context where they would have to adapt their terms to consider them in a risk of bias context. And because our overall goal was to consider all of the different evidence streams in a parallel manner, we asked them to adapt their concepts within a risk of bias approach and then tell us whether or not it was workable. And in the end, we came up with an evaluation of a modification of the standard set of OHAT 10 risk of bias questions, looking at first the applicability, where could we move around various issues, whether specific issues that were not considered and should they be considered outside the tool. And in our attempt, we have all of the considerations fit within that use case in that in vitro risk of bias tool. Here's an example of the adaptation, and I'm just going through one of the questions. I won't go through all of them. But for the first question on the randomization, was the administered dose randomized? Stepping back and looking at what this is for, this helps assure that treatment is not given selectively. And if you look at the text in red, it's not just human subjects and animals, but the concept would be, was it given selectively to individual cells or tissues? And what does it require? That they each had an equal chance, each cell, of being assigned to an individual study group. How does this apply to in vitro studies? If you read down to the blue text, it doesn't apply if you have a homogeneous cell suspension. So if you have a homogeneous cell suspension, by definition, you don't have variation. And so the first question, depending on how you want to approach it, either doesn't apply to that situation, or the answer would be that it has definitely low risk of bias. It's not an issue for that question. And on through the rest of the questions. Now that's focusing on internal validity or risk of bias, but I started by saying there's various aspects to consider within a study appraisal tool, including external validity, the extent to which it addresses the review question or your applicability. And I don't know how many of you might be familiar with the approach in environmental health where after considering the certainty in a body of evidence in order to reach a hazard and a risk decision, the human and animal lines of evidence often come together in a framework like this. This is similar to IARC, but this is what we use in the Office of Health Assessment and Translation. So the human evidence, your certainty in that body of evidence, in this example is rated as moderate, is then considered along with the animal evidence in reaching a hazard conclusion. And you can see how the interaction between the human and animal bodies of evidence helps you reach that decision. And one of the reasons this is because if you consider in observational human studies that you might typically have in toxicology, there would be questions on causation that you couldn't cover. But then you have a whole body of evidence from the experimental animal stream where you could look at various aspects of causation. And so in the risk assessment context, this has been done for a long time to consider those two separate pieces together to answer the overall question. And in this case, the conclusion would be presumed to be hazard based on these two lines of evidence coming together. And then in our method, the in vitro or the mechanistic data together are considered to inform the biological plausibility about whether or not we end up with this overall presumed to be a hazard to humans, or whether we might have even more confidence and reach a known to be hazard, or less confidence and perhaps downgrade it to something else like suspected. And so looking at, in this example of immunotoxicology that I put up there, are there data from the mechanistic studies that would inform biological plausibility of the health effect that you're interested in? Were there changes in cell populations that led to that health effect? Were there changes in the cell-to-cell interactions that supported that health effect? And in this example from the PFOA evaluation where we applied that in vitro study quality tool, the health effect was the development of the suppression of the antibody response to vaccinations. So the cells that are involved in that include B cells, T cells, antigen-presenting cells, and they require the communication of various cytokines and other communication factors. So the mechanistic data that was considered informative for biological plausibility are exactly the data that we have outlined here, and that evidence comes from both in vivo mechanistic studies as well as in vitro studies. So in our evaluation, the assessment of internal validity was not for every single in vitro study that was collected and potentially relevant to immunotoxicity, but rather the assessment of study quality would have approached the mechanistic data, and in this case the in vitro data, that was most relevant for informing the health effect and the biological plausibility. When we're evaluating that evidence, we did it in similar factors to the grade framework: magnitude, dose response, consistency, and what's highlighted here, the internal validity or risk of bias. And then moving on to external validity with the mechanistic data that we have, what these graphs show, the red dots show a significance of the effect, and the blue dotted line is the control value, and if you move to the left, you're moving below the control value at the point that the red dots appear. That's significantly suppressed response relative to the control. And with these mechanistic data, the dose that you were getting, the health effect, suppression of number of B cells, similar for the cytokines, was at a higher level than you were having the effect in the whole animal model. And so in this case, the mechanistic data to consider biological plausibility, yes, there were health effects. Yes, they were of good study quality or internal validity and low risk of bias scores, but the effects were happening at a higher dose in your in vitro models than they were happening in the in vivo animal models, which doesn't support any additional value for the mechanistic data in the in vitro studies. So the external validity was a factor that said that really this data was not that informative. So what are the lessons from our application of this process? It was very satisfying that we were able to evaluate the in vitro study quality along with the others. We had a phased approach. We used to call it an iterative approach, but our language was then corrected by somebody that was more precise. But we have a plan within problem formulation with the proposed use of mechanistic data and including the in vitro studies. We focused in this case on endpoints with relevance to the human health effects that we found. So not all of the mechanistic studies and not all the in vitro studies, only where we found that they were going to be informative. We evaluated the internal study, internal validity in terms of risk of bias using our adapted method, as well as the external validity, which was the factor that really said that this study was, or these data were not as useful in reaching conclusions. And then in evidence integration, we tend to use it for biological plausibility, but the mechanistic data, it depends on your question and the data sets that you find. It may inform lower dose effects, it may inform the relevance of the animal data, etc. So there's multiple factors, and that's why the phased approach to the mechanistic data, depending on what you found, is certainly a method that we would support. All right.
Thank you, Andy.
I don't know if there's any clarifying questions.
Yes, some clarifying questions. We have room for one or two. If you have more content questions, we'll move that to the plenary discussion. Yeah, one from Paul.
Thank you. Oh.
Yeah, you can ask them in a moment. Yeah.
So one of the questions we always have is the external validity of the dose in vitro studies, even when you can get to, presumably in vivo exposure. How do you determine that applicability of the dose
in the in vitro.
When you don't have lower dose studies, right? So you don't know the mechanism that's being active. Higher dose studies is not active. Lower dose studies, when you don't have data, I guess.
So I'm going to paraphrase the question for anybody listening online. Also, I'll define the question I choose to answer. So Andrew was asking about dose and how whether or not the mechanism might be different at higher doses and lower doses, and how you determine whether or not the applicability of the dose in your in vitro studies, and therefore the external validity. I think that's a great question. I think that really in our approach, starting out with determining what health effects data you have in human and animal studies, because it's going to be easier to use and you'll have more confidence in it. Obviously, if we have great human data, we wouldn't even need the animal data. But starting out with the data that you have the most certainty in, and then moving on to the mechanistic data. So in terms of whether you have effects at the same dose, whether you even have internal doses in your human and animal studies, each of these steps would be, what do you have, and then what do you know about it? You would go through what's known about potential mechanisms. In this case, there's a pretty clear mechanism for the antibody response, and it's a consistent mechanism, not multiple mechanisms at different doses, but it would depend on what you have. If you had different things going on and some proposed mechanisms or unknown mechanisms, you'd have to state those and use them in the factor. One answer to your question, it depends. Second answer, it would be a phased approach going through your data. And then the third answer is just to be transparent. Part of the review process is also to see if you missed something. So you would bring in technical experts that might have more information on the mechanistic data or the process involved. And then in the review process, I think you should be prepared for maybe you missed something and to field the information that you have. And the transparency of systematic review puts you in good stead, as does the prescribed approach to grade.
Here you go. I'm Patience as a policy analyst in the test guidelines program at OECD, the Organization for Economic Cooperation and Development in Paris. Please go ahead.
Thank you very much. Excuse me, and thank you to the conference organizers for inviting me. This has really been an interesting morning. So just for a few people who may not be familiar with OECD, I just want to say a word or two about the test guidelines program. I am not actually a regulator, but we are involved at the test guidelines program with developing the harmonized tools for regulatory decision making. So for all OECD member countries, which include Canada, the US, the EU, as well as a variety of other countries around the world, if there is a regulatory requirement for a particular endpoint, they are legally obligated to accept the data produced by an OECD test guideline. Another way of saying that is you test it once in any OECD member country, and all OECD member countries must accept those data. So there's a lot of attention on the tools that are developed to do that, obviously. You've heard a little bit this morning about the AOP knowledge base, which is kind of managed by the OECD, but it's certainly in collaboration with a number of other groups, including the US and the EU. And I'll talk a little bit today about IATA and alternative approaches that are built around AOPs. And then actually I'm going to kind of flip around and talk a little bit about building AOPs around IATAs in some cases. So you've seen at least a version of this figure before. AOPs, I like to think of them as really constructs for organizing data, but they can certainly also be used for developing alternative testing strategies. And when I say alternative testing strategies, I mean strategies that either eliminate or reduce the need to test in an animal, okay? So I think there was a comment this morning that adverse outcome pathways didn't exist 10 or 20 years ago. That's certainly true, although this is really an expansion of concepts that did exist. So it's new terminology that's put on things that have been around for a very long time, things like mode of action frameworks or toxicity pathways, or just sort of the conceptual way that people integrated diverse data to come to a decision. This is much more structured, which allows everybody hopefully to speak the same language, but perhaps not as revolutionary as one might assume from the timeline that's really emerged. So even from the first publication from Gary and company in 2010, the AOPs were really imagined as frameworks for building predictive methods, for integrating testing strategies and guiding the next necessary steps to come to a decision about the chemical safety. And there have been a number of workshops on the fact that that uptake, at least in a regulatory context, has been somewhat limited. As you heard in Michelle's talk this morning, really the AOP wiki is crowdsourced. So what that means is that anybody who's interested in a particular outcome can develop an AOP around that. That doesn't always necessarily mean it has regulatory application. So we're starting to think about a shift in how to increase the utility and the uptake as well. So this is a figure from an OECD guidance document. There's a link to this if the slides are provided. It's a hyperlink. It was also provided to the breakout group. But it's really talking about how AOPs can be used to develop alternative testing strategies. And in this case, it's important to distinguish between the sort of generic AOP here, and you heard Michelle talk about the definitions of each of these terms, the molecular initiating event, the key events, the key event relationships, and the adverse outcome. That is an understanding of the biology behind toxicity. So how the chemical interacts first with a cell or a particular protein, and the sequence of events that lead to adversity. That is different than the tools that are used to measure those effects. So when we think about test guidelines and we think about mining data, what we're really looking at is the in silico, in chemico, in vitro, and in vivo methods that are available here that measure those events. And in many cases, you'll have one test that may include several of those key events. But there's a distinction between the two, and it's important to consider. The other thing to think about is when you have these kind of pathways that start with how the chemical interacts with a cell or a molecule and leads to an organism outcome, you're talking about measuring those effects in very diverse types of tools, very diverse types of assays. So you have to put them together. And the AOP is really very helpful for integrating that information. So an IATA, some of you are going to be very familiar with what an IATA is. Some of you are probably pretty new to this. What this really means is all of the information you can find on a particular type of toxicity, let's say cardiovascular toxicity, put together to determine if you can come to a conclusion. It is an integrated approach to testing and assessment. The assessment is the outcome. The integrated approach is all the different data that you have. So you cast your net, you compile your information, and you organize it in some way. And this is to address a specific problem. Does chemical X cause cardiovascular toxicity, for example? So in gathering all that existing information, obviously systematic literature review is a very helpful tool for that. One determines once they're done gathering the information if one can come to a conclusion. And so that's a weighted evidence assessment or an evidence integration. And an AOP can really help to organize that. But it's important to acknowledge that there are a variety of different ways to do this in terms of gathering your information. There are different search terms and different databases that might be mined. There's a lot of expert judgment involved in not only how you gather the information, but how you organize it, how you weight it, how you consider the strength of it. And so there's potential for very, very different answers from, in some cases, the same information. I think it's important to point out that IATAs, the integrated approaches to testing and assessment, are not one thing, but an actual continuum on a fairly broad scale here of things that include an exceptional amount of expert judgment, or really expert-driven, to something that's much more prescriptive and rule-based. And I'll talk about on that far side here, on the side over here, an example where we're really aiming to remove the expert judgment from the IATA approach. But I'm going to give you 3 examples of how AOPs are used to integrate information in a regulatory context. I come from that bias. Many of you are not so restrictive, but hopefully the things that are true for a regulatory context are also applicable in other contexts that are a little more flexible as well. So this is kind of the gold standard for this type of example. This is the skin sensitization pathway. And this is one case where we have an important regulatory endpoint, skin sensitization. We have a fully reviewed and endorsed AOP. This is kind of the highest level of confidence you can put in an AOP. And all of the key events from the molecule first interacting with the skin itself to the adverse outcome are defined. So on top of that, so here's your hypothetical AOP on the top here. Here's the specific events leading to skin sensitization. But on top of not only having a fully complete AOP with a high level of confidence, we also have a variety of tools to measure each of these key events, and that's really quite exceptional. We have a variety of in chemical and in silico tools, plus a variety of OECD guideline methods, so fairly rigid and high quality methods to measure various in vitro events, sometimes including multiple different key events, more than one here. And we have guideline in vivo methods as well. So there's the local lymph node assay test, which is in mice, and a couple of tests in guinea pigs. And these in vivo tests in particular have been around for a long time. So there's a lot of data here. In addition, skin sensitization is a regulatory requirement for a huge number of chemicals, so that helps as well. So we have a full AOP, we have a lot of tools to measure that, and we have this exceptional amount of data that comes from the Cosmetics Europe Consortium database. And this paper that describes not only the data available, but the curation process on that database was published in 2018 with Sebastian, actually, as the first author, and there's a second companion paper I'll talk about in just a second. But this is truly an unusual circumstance in that we have about 130 chemicals or so for which all chemicals have an in silico prediction of skin sensitization, multiple in vitro methods for skin sensitization, at least one, if not more, guideline studies for skin sensitization, and actually human data as well. So this is really unusual. Okay, so I talked about IATAs, I talked about how they're flexible, I talked about wanting to be on that far right side of the less flexible approaches, and this is what I'm talking about. This is what we call a defined approach. So a defined approach a defined approach, unlike the flexible IATA, uses defined information sources. This is actually a specific defined approach, and I'm not going to get into too much detail on that, but it uses the information from 2 different in vitro methods, plus an in silico prediction. So there's no flexibility in that whatsoever. These are the methods that are required for this defined approach. And it uses a defined data interpretation as well. So this is rule-based. It's a numerical value based on the outcome of these particular information sources. A chemical would be either a strong, weak, or a non-sensitizer, not classified. In other words, any 2 people using this particular defined approach should come to the same answer. We can think about a defined approach as an integrated approach to testing and assessment if you remove all the flexibility. It can be also used in a larger context with more information depending on what your question is. But this is what we're thinking about at OECD to start to harmonize these approaches so they can be used around the world for regulatory decision making. And this is a lot of information. This particular defined approach is from Cosmetics, and it's called the Integrated Testing Strategy, the ITS. But just to show you, bottom line here, if you look at just the sensitivity, specificity, and balanced accuracy of the defined approach against the local lymph node assay test, which is considered the gold standard for toxicology right now for skin sensitization, for the defined approach against the human data for the same values, and the gold standard, the LLNA, against the human data, you see that actually the defined approach outperforms the so-called gold standard test for these particular metrics. So not only are we talking about something that's potentially faster, that's potentially requiring fewer animals for sure, but it actually is a better predictor. Okay, so that's our gold standard. I'm going to step down in terms of how much information we have in the AOPs and talk about a couple of other examples. And this one is from the endocrine disruptor screening and testing context. So even from the initial the publication, the 2010 publication from Gary and others, endocrine disruption was always thought to be a pretty good fit for AOPs because depending on whether it's explicit or implicit, most regulatory contexts, most regulatory definitions for endocrine disruptors require that the chemical has an endocrine mode of action and an adverse outcome in an animal, meaning you're talking about the 2 extreme ends of AOPs. And because those aren't typically measured in the same assay, it requires data integration. And without getting into too much detail, there's a variety of different test methods that are OECD harmonized, a variety of different at EPA, some of which are common to one another. But to come to any conclusion, you need to put this information together. So it's really an integrative process. I'm going to talk about one particular example that's kind of emerged in the last 4 to 5 years or so from the ToxCast high throughput data, and that's the estrogen receptor bioactivity model. So this is taking 18 different in vitro assays, high throughput assays, that detect estrogen receptor interaction at various points in the signaling pathway. This is a real attribute because it overcomes some of the limitations of the particular assays. And because of this integration, you can really distinguish so-called true activity from some of the technology-specific interferences. And not only do you get an estimate of active versus inactive on chemicals, you actually get a value ranging from 0 to 1, which can be interpreted as potency. So in the 18 different assays, we're talking about a number of different biological events, including things like receptor binding, DNA binding, gene expression, and cell proliferation, etc. So we can think of these as being mapped to an AOP, or depending on how you consider an increase in organ weight, perhaps a partial AOP, okay? So you'll notice that we have kind of the same strategy here. We have molecular interactions and cellular responses. I haven't drawn arrows between these to indicate that some of the key event relationships are not perhaps as well worked out. There's a lot of data behind this, but they haven't been vetted to the extent that Michelle talked about in terms of defining key event relationships. But if we look at those different ToxCast assays, those 18 different assays, they basically map to these events. And what we're trying to predict here is the in vivo rodent increase in uterine weight, which is considered a response of estrogenicity. So when we look at how well that bioactivity model performs against a set of in vivo reference chemicals, it really performs quite well, and the difference in the numbers here have to deal with how you deal with things that are inconclusive, that are not clearly 0 or clearly active, but somewhere in the mid-space. Regardless, it does a very good job, to the extent that in 2015, US EPA said that they would actually accept the results of the ER bioactivity model in lieu of having the rodent data in their endocrine screening program. So we at OECD are interested in expanding this sort of, this approach to something that, for chemicals that perhaps don't have ToxCast data associated with them. So how would we take this same thinking and apply it more globally if you have a new chemical or for whatever reason a chemical that didn't go through that ToxCast library? So we've included other assays that might inform on the same endpoints here as the ToxCast one. So the ToxCast are in this pink color. We have some OECD in silico tools in some cases. We have some in vitro methods in some cases. Sorry, these are in vitro methods. This is in vivo. But again, what we're really trying to do is integrate this information to predict organ weight. And this is currently part of a case study that's being evaluated at OECD right now and will likely be proposed as a new defined approach. So in this case, we have AOP thinking. We have AOP, we can call it maybe a partial AOP, that's really informed testing strategies here. And again, same AOP construct above that. And then the last example I'd like to talk about is the key characteristics of carcinogens. We've heard about kind of the IARC approach a couple times in different talks this morning. I want to talk specifically about the key characteristics paper, which Martin Smith's group published in 2016. And this was based on a retrospective evaluation of a variety of chemicals that were known to cause cancer in humans. So you know that from the onset, and then what are the characteristics of those chemicals? Okay, so there are 10 key characteristics. Things like, is a chemical genotoxic? Does it alter DNA repair? Does it induce oxidative stress? Does it modulate receptor-mediated effects? And a variety of other things. This is not to say that these are necessarily causal events, but they are characteristics of the chemicals known to be carcinogens. Okay, so if we map these, and I should say that none of this thinking was done with AOPs in mind. Okay, this was really just a retrospective analysis. But we can still use this kind of AOP construct to organize some of this information. So if we just mapped these 10 key characteristics to various events in a generic AOP pathway, starting with molecular interactions and cell responses and all these other things, we can kind of bin them. And that may be informative at some later date. We are not saying they're causal at this point. We don't have the data behind that. We are not making any assumptions about the key event relationships. This is merely a mapping exercise. And I thought this was so, amazing, and this is a great way to think. And then I went back and read Gary's paper in 2010, and he has almost the same figure. So apparently other people have been thinking about this for a while as well. So how does this key characteristics approach really help to develop integrated approaches to testing and assessment? Well, in fact, these key characteristics are used to identify mechanistic data. And we just heard in the previous talk that mechanistic data can be in vitro studies, can be in silico studies, and can be in vivo biomarkers and things like that. But not only does the key characteristics inform the mechanistic data, IARC has used this approach very successfully in terms of how they integrate evidence, and I think that was also alluded to in the last talk. So IARC uses 3 different streams of data. They use the mechanistic data, which can be from a variety of different assays and in different contexts. They also use experimental animal data, and they use human epidemiological data. The integration of this information, again, can be mapped to this sort of generic AOP thinking without making assumptions about causality here. We still don't know a lot about the key event relationships, but it can still help to come to a regulatory conclusion about the chemical safety. And I'm not going to go into that because I don't have a whole lot of time other than to say, and I believe that that was said in the previous talk as well, the mechanistic information is really most important when you don't have sufficient data in humans. So how might one put all of this thinking together? So first of all, this is an AOP that's in the wiki, but it has not been reviewed and it has not been endorsed, which is to say it's kind of a work in progress. It's tentative. Some of the events aren't worked out. Some of the data is certainly higher quality than others. But it's from David Bewe's group at University of Wisconsin-Madison here. And this is, the title of this paper is, I believe, Personalized in Vitro Screening to Develop Predicted Cancer Outcomes. I mean, so think about that. They're actually trying to design a set of in vitro assays to screen how to treat patients. So this is kind of bringing a number of things that have been discussed in this group together. But you can see that this sort of combines part of that ER signaling partial AOP with that mapping the different evidence to predict cancer outcomes. And this is not a predictive AOP at this point, but it's certainly getting something that's very important and very useful, and it's using that AOP construct to get there. So just to kind of back up and summarize, it's a lot of information at once. There's a lot of concepts thrown in there. But when we really think about how to make use of these AOP constructs, and especially in terms of developing integrated approaches to testing and assessment, whether or not you're trying to predict something that's never been predicted before, whether you're trying to reduce animal use, whether you're trying to do it quicker. All of these 3 approaches that I provided include very diverse types of data that have to be integrated and arrive at a conclusion. In this case, regulatory conclusions, but that's certainly not exclusive. The degree of AOP development underlying the 3 examples is highly variable. We started with a complete AOP and ended with something that has no AOP behind it. And certainly it wouldn't be one AOP, it would be a huge network. And a complete AOP in these particular examples are not needed. And part of the limitation to regulatory uptake in the past may have been the extensive amount of work it takes to develop an AOP. We don't necessarily need a full AOP to make use of it, but we do need data surrounding the key events, surrounding the molecular initiating event and the adverse outcomes. So even for examples that were not built on AOPs, such as the key characteristics, these can be mapped to an AOP framework. I will say, however, that when we're really talking about predictive models, so when we're talking about alternatives to traditional animal testing or the sort of gold standard approaches, these do require a much more complete understanding of the key event and key event relationships, so you need a lot more information underlying there. So these are sort of incremental steps, if you will. So hopefully that's more or less on time. I'd like to just say that this, I've just talked about work from an exceptionally broad number of people and acknowledge those. But also, if you're interested in this, there's a variety of OECD public websites with more information. So thank you.
Yeah, thanks a lot, Patience. So I would like to invite all speakers to come forward. So we're going to open up the general discussion, but start with questions to Patience. So the other speakers, please also come to the floor. And I think I saw someone have a question. Let's see.
Would you like us to be grouped close for body heat?
Yeah, you can use this phone.
Okay.
Microphone. And I have another microphone here. Let's see. Yeah, it works.
It's going to be in
Okay, Danielle Wyckoff, Tox Strategies, thanks. So this is aimed at patients, but I think is really applicable to the entire group. I wondered if you could speak a little bit as to your confidence in the underlying assays as it relates to the confidence in your AOPs. An example of that, I think you gave two good examples with endocrine disruption and the key characteristics of cancer. Endocrine disruption being something we know a lot more about in terms of the pathways, and even having level 4 or 5 assays that are diagnostic of those events. Yet that doesn't, we don't really have that for cancer. So I'm just wondering if you can speak to the importance of the guideline assays as it relates to our confidence, not in the individual study findings, but also as it relates to the ability to predict the outcome.
So that's a great question, and it's a complicated one, I would say. What's considered kind of the gold standard guideline studies are now, there's a lot of work showing that perhaps the animal studies are not terribly predictive of human outcomes in some cases, so it sort of depends what your metric is for confidence. I think one of the things I didn't talk about for time reasons is one of the intended applications of AOPs is for assay development as well, to show where you're missing things. In the case of the key characteristics versus the endocrine example, to me, one of the big differences is for adverse outcomes, there's actually something diagnostic for cancer, whereas for endocrine disruption, that doesn't really exist. You have to make the case that it's an endocrine mode of action, and in general, you can disrupt things like reproductive success from a variety of different mechanisms, including things like calorie restriction. So you're looking at kind of confidence at different ends of the spectrum, if you will. I think one of the ways that the key characteristics have been taken up is to actually look at assay developments. It's certainly in the OECD context, there's looking at a battery of in vitro screening assays for things like non-genotoxic carcinogenicity, so I think that's there. The assumption is that for OECD test guidelines, because they're validated according to a fairly robust process, that's high confidence. But again, I mean, I think some of those, retrospective systematic literature reviews are suggesting that there may be better ways to predict human outcomes. So the answer is sort of, it depends what your metric is and what you're looking at. Did that get there?
Yeah, no, I think that's fair.
Okay.
That's good to understand.
Any other questions to patients?
Or anyone else?
If not to anyone else, I think there were still some open questions.
Maybe an additional comment, and then all of it. So I'll be a little provocative. Is the answer that you just provided just telling us that all of this evidence is indirect evidence helping us draw conclusions in humans? Because I think what you suggested is that some of the animal models are not predictive. I'm saying the obvious, right? But if you try to start putting this into a framework of the certainty assessment, one way of thinking about it is it just informing the question? Another way of thinking about it is, all of that evidence can only be indirect evidence because not all animal models are predictive. Some of the systems that you develop, some of the actual experiments that you develop, they're not going to be never going to be fully predictive. And does that just say, this is indirect evidence, which is good? We're going to use it if it helps. If it doesn't help, if it doesn't predict what we are seeing in humans, then we'll dismiss it. Is that what we're thinking?
Yes. I think all of these are reasonable points. I think ultimately yes, but until you get to something that's better, you sort of need an interim strategy. From a regulatory standpoint, and I know this is a little ridiculous to say, but there is a difference between how well alternative methods predict the animal result versus how well the alternative methods predict the human result. Those are in fact different questions. The regulatory requirements for data are fairly specific and typically animal. So they, obviously we're interested in both if the goal is to protect human health, but you evaluate those separately, as I'm sure that the systematic literature review group knows quite well. So I think what we're looking, until we get some of the really sophisticated, the omics technologies and the 3D tissues and the organs on a chip to a point where they're consistent and predictive of human outcomes, we need something for the short term. So
And one other question, if I may, Paul, is that okay? One other point. With regards to what you showed again, the IARC model for the three different streams of evidence, including the mechanistic evidence. So is there any scenario, I think these are questions that are really important to address, is there any scenario where mechanistic evidence alone would allow you, mechanistic evidence, would allow you to draw strong conclusions? Okay, good. Yes, no, I'm just.
Yeah, so you can't get to there are 3 grades, so there's 4 grades, so, but no, is the
So the answer is no, and is there any disagreement on that? Because I think that's really important.
So is it no that we're not going to get there? Because we're trying to look at the mechanistic data, and some of your statements about everything that you talked about means it's all indirect. So in the framework of considering the IARC framework and other human risk assessment or hazard assessment frameworks, it's bringing those different pieces of information together for the value that they have. And so it's indirect. If it was to stand alone, you can only reach a certain conclusion. But if there was a case like the LLNA and the skin sensitivity is this wonderful example, and it may be entirely unique or partially unique about the amount of information, but also the biology that's captured in that pathway, all of those things. In those cases, the mechanistic data could get you there, but it's built on top of all different other things. So I'm not quite saying yes.
But that means no, because I think this is.
But also the goal and the aspirational is to see what would it take to get there, because we want to develop the frameworks that would get us there.
That's exactly the reason for why I'm asking, right? I think one way of starting to address that in the context of the certainty assessment in grade is to say, what are the goalposts, right? So there may be exceptions to that, but and to work through scenarios, let's just say there is one human case where there is extremes are good, where there is a relationship between sub-exposure and a cancer that was observed. Okay, that's all that you have, because there's always human evidence, otherwise you wouldn't, or animal evidence, otherwise you wouldn't be asking these questions. And now you have very strong mechanistic evidence that people put together for that extreme case. Would that allow you to draw strong conclusions? And the answer is probably no, but you will end up somewhere,
how rare is
low certainty.
How rare is the cancer?
Sorry?
How rare is the cancer? You had the one study.
So that may influence it. But even if it's a rare cancer and you have one single case, will you ever end up? Yeah, will you ever end up drawing strong conclusions? And the answer is probably no. You need a little bit more, which tells you, is that with the mechanistic evidence alone, you cannot reach these very strong conclusions or high certainty in the exposure outcome relationship. Maybe, I don't know.
I don't know that I would say that necessarily. I mean, it's a high bar that you've set, there's no question, but I think right now there are a variety of groups that are working on trying to predict human cancers based on mechanistic data alone. And it certainly helps to have a lot of data that are integrated, a lot of mechanistic data, and develop these kind of screening batteries along with things that you think are essentially sequelae in a sequence of events. So you're talking about something that's orthogonal, essentially. Here's the first step, here's the second step, here's the third step. But, I mean, I think that's the current goal, and I do think we'll probably get there, but it's going to take a better understanding of what happens in the underlying biology.
I apologize, Paul. No.
So I
do want to disagree and say there are conditions where you could get to strong based on just mechanistic, because I think we're lumping mechanistic evidence too broadly into just predictive, mechanistic in vitro toxicology, because you could have a very predictive biomarker in humans that we would consider mechanistic evidence, like pleural plaques. And if you were seeing a chemical cause in pleural plaques, you would say, I presume this is going to cause cancer. And that would be a linkage in terms of the biomarker already being linked to a particular cancer with another chemical. You would then be strongly certain that your particular chemical is causing that same cancer based on previous understanding of the biological linkage. But that's not, I think that's not what you're asking. But I'm saying that we're lumping mechanistic evidence too broadly into this, or too narrowly into just predictive molecular toxicology.
No, I was referring to the AOPs, the models that were shown here, because in your case, there's indirect evidence from other populations, right, that you would utilize on humans, that you would utilize in all these conclusions.
But not on your chemical, it'll be on another chemical, but indirect.
Yeah.
Well, and IARC does distinguish mechanistic data in exposed humans, but they still require another source of information to get to the category.
Well, I just started because I thought there was no question.
Go, Paul.
So I was just trying to not drop this. So Paul Whaley, EBTC in Lancaster University. Andy, I had a question for you about your search strategy and how you kind of set your boundaries for inclusion criteria for the immunotoxicity mechanistic evidence, right? Because if we think about, so from what I understand of the AOP stuff that's been presented this morning, you could kind of end up going quite a long way upstream on the sort of intermediate endpoints that haven't necessarily been investigated for the chemical you're interested in, but because they cause the outcomes that you're interested in, would still be relevant to your review. So how did you handle that in your systematic review?
So that's a good question. So the way that I'm interpreting the question and what I will answer, again, was in the search strategy for the immunotox risk assessment of PFOA and PFOS, how did we define the search terms to capture the mechanistic data? And we consulted the immunotox experts. And in immunotoxicity, there's a lot of in vitro signals and potential mechanisms for many of the phenotypic events, but none of them are really panning out or have panned out to this point as being predictive without multiple events along a pathway all being shown to be perturbed in order to produce that. They're good as biomarkers for now you should follow up with additional studies, but they're lousy for being predictive of the actual immune effects. So we were not that careful to capture the mechanistic information in that first literature search. However, we subsequently went back when we focused on the antibody response to look at the cell types and the communication types that we knew, according to the literature and the experts, were involved in producing those immune functions. So the first one was not that all-encompassing, and the second search as a follow-up for those mechanisms was.
Okay, so because it was kind of a little bit about inclusion criteria as well, so you kind of had this iterated approach where you had...
Phased approach.
Is it phased? Sorry, I thought, so is it the phased the correct term, not iterated? Phased, okay, right, sorry. So you had this phased approach, so you kind of did a follow-up review of a pool of the mechanistic data.
Yeah, so
you didn't have like a great big database of all the mechanistic stuff and then some inclusion criteria.
No, and we would not recommend that.
No, it sounds like a huge...
If you have a evidence approach where you can use automation or you have a large number of people that don't have anything to do, you can collect all that information, but I challenge you on how useful the information will be unless your plan is that you want to gather all the mechanistic information and then try to sort through it to see if it's starting to support some of these key characteristics and how are you going to use it. But if you're really making the decisions on the end of the health effects, we would suggest that you start with that health effects in humans and animals and then dig deeper where necessary. If you can capture the mechanistic data along the way and bin it as potentially relevant later, but don't spend a lot of time in pulling all the information out, evaluating study quality, contacting the authors to see if they missed something, for something you're never going to use.
Okay, can I be greedy and have a follow-up and pull Michelle in as well then? So if you've got a situation where you have a potentially very broad evidence base that seems applicable, but you don't have the resources at the point of asking the question to kind of really get into that, would large evidence maps where someone's already kind of sketched out the topography of that evidence base that you could then query in the course of maybe setting up a systematic review, would that be helpful for identifying potentially relevant mechanistic information?
It would be fantastic if the literature, as it was being published, besides the fact that as we publish literature, you have data, and then in order to get it published, you strip out some of the information and put it in this PDF format that then somebody else has to try to get the data back out. So it would be nice if we were all dealing with the original data. But if somebody were out there collecting data on exposures of everything as it was published, potential animal species, strains, models, health effects, and that it could be grouped by machine learning, artificial intelligence, and then we could start to make decisions on top of those groupings, that would be fantastic. And we're probably not that far off. That far off is 10, 20 years, I don't know, but until we get to that point, if somebody else did those efforts, you could follow up where there were pockets, where there were gaps, where there was novel associations that you hadn't seen before. I think that we're on the cusp of all of those things.
So if I can butt in real quick. I think that, Paul, when you were asking about some of the information, and Andy, when you mentioned that it's maybe not so useful, a lot of that information is really important for the relationship between events, right? So how do you get from an event to another? And maybe the information that you're extracting were not key events, but it's really important for connecting the dots, essentially. And so then what you just mentioned as far as, you know, the evidence mapping and getting all the information extracted and mapped, whatnot, that's kind of the beauty of the AOP paradigm, is that it does provide a like an informational framework for capturing the information that we're actually extracting. The challenge is getting those relationships identified, right? So we have all this information, and there's a lot of different pieces, but we don't know how they're related to each other. And so I had a conversation recently with a cardiologist who mentioned that they were doing a study and they only reported on one biomarker where they had a lot of other, useful information that they measured from humans, but they didn't report it because they didn't see how it was relevant. But if you begin to take that information and align it in your framework, you may identify, pathways or relationships or relevancies that otherwise you didn't know existed. And so as far as getting back to the mechanistic information kind of supporting our particular outcome, therein lies the beauty in that, if we can get all this information harmonized into a common space, such as the AOP framework, from this rich literature source, that we have in existence and are extracting the data from, we would be able to populate it so that, all the partners could begin interrogating that for information relevant to their particular question. It's just that we're constrained by resources, and we're constrained by, how the information and data are presented to us in the PDFs, because we have people collecting it, putting it into a database, and then spitting it back out into a PDF that we can't have our machines read very easily, and then there's content in there that we want to extract, but again, it's in words, right? The terms are all different, so it makes it hard for people like you to do your jobs to get at it, so that people like us can actually use it. And then people like you, again, make the algorithms to query, interrogate that information base. So yeah, it's kind of like the circular thing, but I think with time we'll get there.
While we were waiting. So, but your biomarker example was, while we were waiting, the biomarker example was a good example for where the information that you may be collecting informs you about how direct the actual endpoint that you're measuring is connected to the human outcome, right? So that's a perfect example for thinking about indirectness, validating a biomarker, because, in some instances you wouldn't do it. HIV viral load, for instance, and the outcomes related to HIV. You don't need to look at much more mechanism. You know that these things are related. In other scenarios, as you described, you measure a biomarker, but you have no idea how it relates to it. You will only validate it at the end by looking at the health outcomes, but the type of evidence that you're describing may help you for the time being to say this is a good surrogate, still being concerned, or it's a bad surrogate, right? So that falls directly into the indirectness of the outcome category. And I think it's really important to categorize that. Where are we? So one is trying to establish the pathway. That's all fine. And the other is trying to identify in what part of the exposure question you have confidence. In this case, it's the confidence in the outcome. Anyways.
Hi. I have a question sort of on a different theme. This might be more for you and Jan. So thinking about grade, and over the past couple of years, this idea about mapping to the indirectness has come up a lot. So what is your current thinking, and maybe more in the brainstorming mode? I know we need case examples, but when you're trying to envision a scenario for trying to sort of describe this, where you've got this mechanistic information, and let's just say it's both high throughput screening, it's experimental in vitro studies, it's animal-based. And how would you kind of think about describing the directness when you've got diversity? And I'm thinking about some of the approaches that have been put forth for putting randomized controls and sort of observational studies on the equal playing field. So how would you how do you envision that happening, kind of also maybe recognizing that we don't have a risk of bias tool that would put that on common footing right now?
You want to start again, or should I start? Okay, very quick question. I think it comes back to some of the conversation that we had and what we tried to visualize. I do really think at the end we are interested, if we're talking about human health, we are interested in the question, the population being humans. I repeat what I say repeatedly, right? So if we just agree, if we're in the veterinary space, our population may be animals, right? But right now we're talking about humans, and it's the issue of humans, exposure, and outcome. And I think the mechanistic evidence informs extrapolation in these various spaces, as we just said, from animals to humans. The mechanistic evidence will tell you, for instance, there is a pathway that is similar in the guinea pigs and in the humans, and we only have guinea pig evidence, but we may not be concerned that it's only guinea pig evidence because the mechanistic evidence supports that the pathway or the mechanism is actually the same in humans. And the same, I think, is true for the exposures and the outcomes. That's really where this falls in. It falls into this indirectness category, and that's where my question was coming from. I think it will be extremely difficult to draw strong conclusions unless you have some of this indirect human evidence or direct human evidence, and that's, I think, where the indirect evidence right now falls into. And it's really extremely helpful, right? It can be extremely informative for the various aspects. But where I think we continue to struggle is that scenario that we described, basically no human or animal evidence, and you just come up with a pathway. I would say that based on Andrew's point, I think there always will be indirect evidence, even for that path, even for relating the pathway to human evidence. But that's where then you draw these conclusions about these three elements.
Rob, you're going to have another question.
I have a question, actually, a clarification of a total ignorant in AOPs. I understand that in order to make any predictions in humans, we would have to know the pathway in humans and in the whatever system we are actually measuring. So you need to know lots of these pathways and make sure that they are the same, because they may be very different.
What's kind of funny about that is, maybe not so funny, we already do know a lot of these pathways. I mean, biology is biology. You perturb it, based on an environmental exposure, and some things will still be the same, right? So we have a ton of mechanistic information about how, like KEGG pathway, how energy is metabolized, how energy is produced, how electricity is conducted. So we have a lot of this type of information, and we have a lot of connections already to diseases. So I think that another good one, Steve, I'm thinking of you because there's the Monarch Initiative, which has the disease phenotypes and the connections to different pathways. So there are a lot of resources. We do have a lot of information about these different pathways, mechanisms, modes of action that lead to disease. And, again, biology being biology, we can kind of take what we already know from the medical community and what we've learned about humans over the past, thousands of years and kind of apply that in the toxicology space.
That's a much better answer than what I'm going to give. But what I will say is that right now, the in vivo animal studies are assumed to be relevant to humans with no real data supporting that. So you don't necessarily need, what you need is information that will replace the current standard for testing for human health. And right now, that's predicting the outcome in an animal. That is certainly something that is kept well in mind for predictive models to be human relevant, and there are lots of ways to increase the human relevance, but that's actually... How much of the high throughput is in human cells? Quite a bit. So a lot of the high throughput is in human cells, as Amy stated, and there are certainly, in terms of the omics, there's a lot of information behind, what pathways are activated and things like that. But in fact, to replace the animal data endpoint, what you need to do is predict the animal data outcome. For now, hopefully something better than that.
Yeah, I think that was my question. It was more generic. It's just if you're predicting human, animal, this animal or the other animal, because fish or rabbit or I don't know, whatever else, bacterium outcomes. The question is if the bacteria, if the pathway in bacteria is similar to pathway in rabbits, is similar to pathway in fish, et cetera, because there may be some other biology, but, so in order to be able in some other species or some other system to predict what happens with the rabbit, which is current gold standard for assessing that, I'm making it up, but, then you need to know both what happens in the rabbit and what happens in the other thing, and that they are similar. Okay. So well.
Yeah, thanks. This might be a good time to wrap up before lunch. Of course, you can continue the discussions during lunch. Let me start by thanking all the speakers of this morning and giving a small gift.